Assalamualaikum and good morning to both my panels and fellow friends. My name is Wanushara Auni binti Wan Zakaria. Today I am delighted to present my final year project titled Detection of mecA gene in coagulase negative Staphylococcus isolates from subclinical bovine mastitis contaminated raw meat sample under the guidance of my supervisor, Madam Rafida binti Rasul. Today I will be discussing about coagulase negative Staphylococcus or CoNS, which is significant pathogens in mastitis. This bacteria oftenly carried mecA gene, which confers resistance to methicillin and other beta-lactams antibiotic by encoding a penicillin-binding protein called Pb2A. The research addresses two key questions. What is the antibiotic resistant pattern of CoNS isolate from subclinical bovine mastitis milk sample, and do these isolates possess the mecA gene contributing to their resistance? The objectives are to perform antibiotic susceptibility tests on these isolates and to investigate the presence of the mecA gene using PCR. This study is crucial for improving health management in dairy cattle, particularly in Malaysia. Literature review. The mecA gene in bacteria produce Pb2A, which reduces the effectiveness of beta-lactam antibiotics. Beta-lactam normally inhibit penicillin-binding protein, which is involved in bacterial cell wall synthesis. For Objective 1, aim to perform an antibiotic susceptibility test on CoNS isolates obtained from subclinical bovine mastitis milk samples. The process began with the revival of the obtained isolates, followed by the growing of sample on specific media. After adequate growth, antibiotic susceptibility test was conducted to determine the resistant profile of each isolates. Moving to the second objective, the goal was to investigate the presence or absence of mecA gene in the CoNS isolates using PCR. Initially, overnight growth sample has been prepared before the DNA extract using DNA extraction by Pure genomic DNA kits. The DNA was analyzed for purify, followed by amplification of the mecA gene using Fyp2 master mix and specific primer for each gene. The slide shows the morphological characteristic of CoNS on mannitol salt agar (MSA). On the left, there is clean sample with round-shaped colonies that appear cream-colored with smooth edges, and the MSA remain pink. CoNS typically does not ferment mannitol, so the medium does not change in color. On the right, the contaminated sample shows golden yellow colonies with a yellow medium, indicating mannitol fermentation, which is typically for Staphylococcus aureus, and it is not CoNS. This observation helped to differentiate CoNS from Staphylococcus species. Next, the slides present the result of antimicrobial susceptibility test for CoNS isolates. The table shows that susceptibility of each isolates to various antibiotics. The most susceptible antibiotics are tetracycline, gentamicin, and erythromycin, followed by ampicillin and vancomycin. The highest sensitivity is observed for penicillin and oxacillin. Identified multi-drug resistant samples include UMMP 66, 84, and 222. On this slide, we discuss the mechanism of beta-lactams antibiotics, specifically penicillin and oxacillin, which are part of the beta-lactam class. These antibiotics typically work by inhibiting penicillin-binding protein, crucial for bacterial cell wall synthesis. However, in CoNS, isolates that carry the mecA gene, resistance is mediated through the production of Pb2A, an altered penicillin-binding protein. Pb2A reduces the effectiveness of beta-lactam antibiotics, including penicillin and oxacillin, by not binding to these drugs. As a result, bacteria can continue synthesizing their cell walls even in the presence of the antibiotics, leading to resistance. Our study found that the highest resistance rates for penicillin and oxacillin, highlighting the presence of mecA-mediated resistance in these isolates. This slide shows the result of DNA extraction using the Pure genomic DNA kit. The presence of band for all samples indicates successful extraction of DNA. However, some faint bands were observed. This could be due to low DNA concentration or partial degradation, possibly caused by incomplete cell lysis, inefficient DNA recovery, or improper sample handling. Despite these issues, the extraction was largely successful, allowing to proceed with further analysis. There were 6 trials for the PCR test with various troubleshooting attempts. Despite multiple efforts to adjust the sample amount, annealing temperature, and refine the process creation, the PCR condition for detecting the mecA gene proved challenging and couldn't be fully optimized within the project timeframes. This highlights the complexity of the optimization process and the challenges encountered. Unfortunately, further optimization wasn't possible due to time constraints. For the first troubleshooting step, we increased the sample volume to increase the DNA concentration in each sample. This was necessary because higher DNA concentration can improve the efficiency and accuracy of PCR amplification, leading to more reliable results. The table shows the adjustment made to the annealing temperatures starting at 58.5 degrees Celsius for the first 3 trials. To enhance primer binding, the annealing temperatures were lowered in subsequent trials down to 51.8 degrees Celsius by the 6th trial. Despite this adjustment, the desired results were not achieved too. So, several possible issues could explain the absence of bands in this PCR result. First, the mecA gene might be absent in these isolates or present in very low copy of numbers that are below the detection limit of PCR assay. Additionally, the DNA quality from extraction can significantly affect PCR efficiency. If the DNA is degraded or impure, the PCR reaction might not work as expected. Lastly, it's possible that the reagents such as tag polymerase, MgCl2, or buffer have degraded due to multiple freeze-thaw cycles. Degraded reagents can lose their effectiveness, leading to poor or no amplification. These factors combined could have contributed to the lack of detectable bands in these experiments. In conclusion, the study aimed to detect the mecA gene in CoNS isolate from subclinical bovine mastitis milk samples. Despite rigorous PCR optimization and DNA extraction, consistent mecA gene amplification wasn't achieved, suggesting challenges with resistant mechanism or gene absence. Moving forward, it's recommended to use fresh reagents, minimize freeze-thaw cycles, and optimize PCR condition. Additionally, exploring alternative detection methods like the cefoxitin test could provide valuable insights. This method involves using cefoxitin to screen for mecA-mediated methicillin resistance, offering a reliable tool for detecting resistant staphylococci in veterinary settings.